Hello everyone, I made this video to help you out with protein synthesis. It's literally going to walk you through the process. It should be very simple. If you found it kind of confusing in class, please look at this video as many times as you need to. It's right to the point. So let's get started. Alright, so what's the purpose of protein synthesis? Synthesis, you know what that means. That means to make something. Protein synthesis, to make proteins. So the whole purpose is to make proteins for the body, period. Now, more specifically, it's to get the instructions for proteins to the place where proteins are made. It's still pretty simple, okay? If you're going to make proteins, you need instructions for them. So that's the whole purpose, is to get the instructions for proteins to the place where they make proteins. Here's the steps we're going to take. Number one, we're going to convert DNA to mRNA, and we're going to do that by using the base pairs. Let me see if I can actually, I'll try it. Sorry about that. Okay, yep, I think it'll work. I will show you the base pairs real quick. And please forgive me because I'm writing with my finger. So let's see how this works. Base pairs, remember when you're dealing with DNA, whoa, like a kindergartner. A, adenine will always pair up with T, thymine, and then G, guanine. That's supposed to be a G, everyone. G will always pair up with C. That's when you're dealing with DNA. All right. Now, when you're dealing with RNA, which is what we're going to be using, because it says here we are converting DNA to mRNA, so the base pairs will be slightly different. A will pair with U, uracil, and then G will still pair with C, cytosine. Okay, that's the only difference. All right, going back to step number 2, we're going to put the codons from the mRNA into the codon chart. You all should have seen the codon chart by now, and later on this PowerPoint there is another chart, so we'll show you how to use that. Number 3, last step, it says to write the amino acids from the chart, which will make your protein chain. All right, so pretty simple. Here's the 3 steps again. We're going to convert DNA to mRNA by using these base pairs right here. And then number 2, we're going to put the codons, which are those 3 base pairs of the 3 bases of mRNA, into the codon chart. And then from the codon chart, we're going to write down the amino acids from the chart, which will make our protein. Let's move on. All right, so here is your original DNA strand. This is what will be given to you on any worksheet, on any type of problem that has you doing protein synthesis. They will give you the DNA strand. So the DNA strand, CCG, TAG, GTA, ATT. All right. First thing we need to do is get the RNA strand. And we do this, again, by using the base pairs. Okay? These base pairs right here. Going to use those. All right. So, what pairs up with C? That's what I want to know. What pairs up with C, C, G? All right. From the base pairs, we know that it's going to be G, G, and C. All right. Now, what pairs up with T, A, G? It's going to be A. Remember, with RNA, since we're writing RNA, you're going to write U and then C. And then again, for G, you'll write C. The T is going to give you what? I'm waiting on you to tell me. I'm not going to write it down. No, just kidding. It's a video. I am going to write it down. A. And then for A, you'll have U. A again, we'll have U. TT will give us A. So that will be our full RNA strand. All right. Next, we need to find the amino acids. What goes here, 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 and here. And that is when we do translation, and we're going to actually use the codon chart. Okay? So the process going from DNA to RNA is transcription. The process of going from RNA to the amino acid, aka to the protein, is called translation. All right, let's move on. So we're going to take these codons right here, and we're going to put them in the codon chart. Here's your codon chart, and up here at the top, you have your codons from the screen beforehand. So let's take the first one. G, G, C. We take that, we put it in here. You want to find the largest G. Where is the largest G? It's in the top top left quadrant. So you go G, then you find the next biggest G, G, and then you go down to C, which is right here. And then you're going to write this amino acid, GLY. So we'll write that GLY for our first amino acid. And now we're going to start with this one. That was the first one. We're going to start with this one. AUC. So we're going to go A. Oops, my bad. We're going to go A, U, and then C. And that'll give us ISO. ISO, ISO. And then we're going to start with this one right here. CAU. You look for the biggest C. C for the first base. Second base, A. And then U right here. I'm doing different signs so that way you can tell the difference of when I match them up. And that's going to be HIS, H-I-S. Lastly, UAA. Should be pretty easy to do. UAA. And it says stop. So that's where we would stop at. That's our stop codon. All right. This is our protein chain. We just got it earlier. GLY, ISO, HIS, and, ooh, hold up. This may not be correct. Let me go back and check. Is UAA, UAA. Yes, that's not correct. It should be a stop codon. Let's back that up. All right, glad we caught that. So that is not correct. It should be a stop codon. Good. I'm glad we caught that. If that had happened and the LEU was made there on accident, just like we did it, that would be a mutation, and it would make a different protein. Kind of like, this is an extreme example, but if you cut your arm and you needed new skin to go there, and you needed the same exact skin that was there beforehand, your body is supposed to code for skin. So you're making skin cells. If you have an error just like we just did, that's called a mutation, and you might make a hair cell instead, or make a nail cell instead, and then you have a nail in the middle of your arm, and you look very, very weird. All right, so getting back to the PowerPoint, the chain of amino acids from the codon chart is a polypeptide. So that thing right here, that's a polypeptide, which is the same thing as a protein. Okay, pretty good. So here's your end result. At the beginning, we started with this DNA strand right here, which is going to be the template strand. It's from the original DNA. We split it, and then this mRNA came, these free nucleotides, came in and made a copy. This is the RNA copy of that DNA strand. And then once we put it into the codon chart, we got out these amino acids right here. And they're all linked together by those peptide bonds. And that would give us our protein. All right, and remember, we said the LEU was an error. It's supposed to be a stop codon. Okay? And that means just what it says, stop. It's telling your body when to stop making that protein. All right, guys, I hope this helped. And until next time, this is Mr. Macon signing off.